Hello, and let's start with a new topic, cancer. So basically, I have made this diagram of cell cycle. You can see these are the stages of cell cycle: G1, S, G2, and M phase. This is the first gap, and S is the DNA replication phase. G2 is second gap, where this gap is required for making a lot of enzymes which are required during meiosis or mitosis. So I have made this cell cycle because I wanted to show you here. See, checkpoints. These are the checkpoints which act as regulators. They may be inhibitors or they can be activators. They basically do cell cycle control. So the problem with cancer is that they do not follow these cell cycle control points. Okay, so they basically can cheat these regulators. So what is cancer? It's a disease of uncontrolled cell division. Uncontrolled cell division means cells will keep on growing. They'll keep on growing irrespective they have been regulated or not. So now its development and progression are usually linked to a series of changes in the activity of cell cycle regulators. See here you can see the difference. This is a normal cell. Let's assume this is a Petri plate and you're trying to grow some normal healthy cells. These are the normal cells and they'll keep on growing. They'll keep on growing and they'll keep till a point they start contacting each other. Once they contact each other, they will not overcrowd and they will stop inhibiting each other and they'll stop growing. This is called contact inhibition. Okay, so contact inhibition basically means when they contact each other, they stop growing, they inhibit. Growth is inhibited. These are the normal healthy cells. In case of cancer cells, you can see they do not have any contact inhibition. So loss of contact inhibition, this is the first property of cancer cells. They keep on growing, keep on growing, irrespective they are touching each other or they are in contact with each other. So there is a difference between tumor and cancer, which normally people get confused with. This is a normal cell. You can see the size. A normal cell is very small in size, right? Where if it's cell, this cell becomes enlarged. Okay, so the enlarged cell, a cell which is enlarged but it is localized. Localized means it's not spreading. It's immobile and it do not metastasize, right? So, but cancer cells, they are not localized. They can cross the boundaries and they can go to different locations. Now, difference between cancer cell and normal cell. So first is that cancer cells can multiply and culture without any growth factors or growth stimulating protein signals being added, whereas normal cells, they need growth factors to grow in culture. This is the same thing which we discussed. See, the normal cells, in presence of growth factor, they are dividing. In absence, they are not dividing. Cancer cells, irrespective of the fact that growth factors are there or not, they are still dividing. Second property, which we again discussed, that cancer cells ignore signals that should cause them to stop dividing. So they do not follow contact inhibition. So that means loss of contact inhibition in case of cancer cells. Whereas normal cells, they have contact inhibition. Third point, cancer cells can divide many more times than a normal cell of the body. This property is called replicative immortality. So these tips of the chromosomes, they're called telomeres. So what happens after each cell division? Telomere shortening takes place. Okay, so these telomeres, they shorten up and this is called aging. This is how a cell ages. Now cancer cells, they can express a protein which is called telomerase. Telomerase is a protein, is an enzyme which is expressed by the cancer cells. What these enzymes does, they reverse the shortening of chromosome ends that normally happens during each cell division. So this is their fourth property. Fifth, cancer cells have the ability to migrate to other parts of the body, and this process is called metastasis. And to promote growth of new blood vessels, there is a process called angiogenesis. Everything is helping them in growing. So angiogenesis, basically it gives tumor cells a source of oxygen and nutrients so that they can grow properly. Cancer cells also fail to undergo programmed cell death. Now what is programmed cell death? Programmed cell death is apoptosis. Apoptosis basically is a process which normal cells, they follow. Whenever normal cells they die, so they die in a programmed way. Okay, so this is how a cancer develops. This is a normal cell. Now the development of cancer can be due to many things. The one of this is mutation. It can be exposure to a radiation. It can be exposure to a carcinogen. So when a normal cell converts itself into cancer cell, that the major reason behind it is mutation. And most cancer cells arise because of mutation that make them divide quickly. These are benign tumor, a mass of cell that divide too much but don't have potential to invade other tissues. Okay, this is the definition of metastasize. Now, a group of cells that divide excessively and can invade other tissues, they are called malignant tumors. Right? So now a benign tumor converting itself into a malignant tumor. So this is all about cancer.